Good morning, sir. This is Devan Chopra, and I am here to present my GD on the topic Fermentor Design and Types of Fermenters. So, before we go ahead with your topic, let us have a brief introduction about fermenters. So, fermenters refers to a device that provides all the basic necessities important for biological product extraction. A fermenter contains different devices that helps to maintain the environmental factor inside it, which in turn lead to the production of biological products. Therefore, the main objective of a fermenter is to maintain a controlled environment that supports the growth of the bacteria or any other organism. Okay, so in simple words, fermenter provide aseptic condition for an organism to grow in it. So this is a diagram of fermenter. In this we can see various components of a fermenters. So the main parts of fermenters are temperature control sensor, agitator or impeller, baffles, aeration system, asperger, pH control sensor or foam controlling device. So these are the parts of fermenters. So now we will discuss about each of the part in brief. So temperature control sensor. As we all know, microbes grow in a particular temperature. So a sensor is provided at the top side of a fermenter to keep a track on the temperature inside the fermenter. And we can see about it in the previous image which I have used. Here it is, the sensor. Okay, now agitator. Agitator or impeller mixes the nutrient media so that each and every cell gets equal amount of nutrition. It also prevents the settling down of nutrients. This impeller is connected to the motor. So impeller or agitator helps in the mixing of the material so that every cell get the equal amount of nutrients, and it also prevents settling down of nutrients. This is connected to a motor for continuous running. Baffles. It helps in the prevention of vortex formation. So baffles are side mounted walls which help in the prevention of formation of vortex. Vortex can be defined as a tornado which gets generated inside a fermenter. So baffles help in the prevention of formation of vortex. In simple words, we can say that एक भंवर सा बनने से रोकता है baffles. So now aeration system or asperger. So it is a hollow plate which lies in the bottom of the fermenter. It consists of two plates, the top one which has holes in it and the bottom one through which the oxygen passes to the top plate and it comes out in the form of bubbles, and the bubbles are then broken by impeller to release the oxygen inside the bubble. So now next one is pH control sensor. So as we all know, microbes grow only in a particular pH as well. So there are some sensors which are also present on the fermenter to keep the track of the pH inside the fermenter. So if the pH drops below a certain point, the pH sensor indicates that the pH is going down, and it automatically maintains the pH inside the fermenter. The last one is foam controlling device. It's a device which helps in the prevention of foam. So as we all know, as the impeller will move, so there might be a chances that foam formation take place. So that's why there is a foam controlling device. It as detects the foam is being generated, it get activated and helps in the prevention of foam. So now we will discuss about types of fermenters. So there are multiple types of fermenter being used, but I have taken the example of few fermenters, and they are continuous stirred fermenter, bubble column fermenter, fluidized bed fermenter, and the last one is airlift fermenter. So first we will discuss about continuous stirred fermenter. So it consists of a cylindrical vessel which is occupied with a motor driven central shaft which support one or more impellers. Shaft is present at the bottom of the bioreactor. The air provided to the culture medium is through the asperger. And advantage of stirred tank fermenters are that they provide efficient gas transfer for the growing cells. So as per the name suggests, continuous stirred fermenter, in this continuous stirring of fermenter is done, and the impeller is present at the bottom of the fermenter, which helps in better mixing of the gas in the fermenter through, which comes through the spreader. So this is the advantage of continuous assisted fermenter. So now we will discuss about bubble column fermenter. The bioreactor involves agitation by densely driven fluid molecules, and the sparger is used to provide air into the continuous liquid phase at the bottom of the reactors. The fluid mixes intensively at the high gas flow rates when convective flow becomes turbulent. So in this, the densely driven fluid molecules are mixed by the help of air bubbles which comes from the sparger, which is located at the bottom of the fermenter. So now we will discuss about the third type, fluidized bed fermenter. It is almost same as bubble column fermenter, but in this the upper part is a lot wider. So now we will have a look at the definition. This type of reactor is used to carry out multi-phase reaction. The fluid passes through a solid material at high velocities to suspend the solid. It provides the excellent mixing, increased mass transfer and enlarged surface area, which increases the efficiency. Removal of heat and exchange of gases are easy. It requires more pumping power, and also chances for erosion of internal components are more. So in this the chances of erosion of internal components are more, but it provides excellent mixing and increased mass transfer and enlarged surface area, which increases the efficiency of mixing in the fermenter. The last type is airlift fermenter. So the medium of the vessel consists of a baffle or a draft tube through which air is pumped. This might create a bubble in the medium, which ultimately helps in rising up through the baffle tube and drags the surrounding fluid as well. This, thus, in this process, still up the contents by air. There are two types of airlift bioreactors. The first one is internal loop type, and the second one is external loop type. So, in internal loop type airlift bioreactor, single container with a draft carrying out fermentation process, and in external loop airlift bioreactor has an external loop separated samples or liquid in a different channel carrying out fermentation. So we will have a look at the airlift fermenter through diagrams. So in first one is the internal loop airlift fermenter. In this we can see there are loops inside the fermenter in the same cavity, and in the second diagram we can see that there is a different loop on the right hand side to carry out fer separation of samples. So that's all from my side. Thank you and have a nice day. Bye bye.